Hi everyone, and welcome to another episode of Investigating Rare Diseases. I'm your host Isabel Anderson, and today we are going to be talking to my good friend Rachel. Rachel's son was recently diagnosed with a rare genetic disease called Tay-Sachs. So today we will be talking to her about that and answering any questions she may have. Hi Rachel, and thanks for joining me today.
Hi Isabel, thanks for having me.
First, I'm sorry to hear about your son's diagnosis. I'm happy to answer any questions you may have today. Before we start though, to give our listeners a little bit of background information, Tay-Sachs is a rare genetic disease caused by a mutation in the HEX-A gene. The mutation in this gene causes a deficient amount of protein that is needed to break down fatty substances. The symptoms associated with this disease are loss of motor function, muscle weakness, seizures, and a loss of mental functions. In infantile form of this disease, which is more severe, we also see respiratory and cardiac abnormalities.
So, since the symptoms of Tay-Sachs are loss of mental and muscle functions, does that mean neurons and muscle cells are affected in this disorder?
Yes, Rachel, you're exactly right. In Tay-Sachs, there is a buildup of these fatty substances in primarily neurons, but we also see this buildup in muscle cells. And so when we see studies being done on Tay-Sachs, usually neurological cells are the ones taken.
Okay, that makes sense. So, my first question for you is, what is the protein involved in Tay-Sachs?
The protein involved in Tay-Sachs is called beta-hexoamidase subunit alpha. So earlier I mentioned that the gene HEX-A is mutated in Tay-Sachs, and that affects the transcription of the protein hexoamidase subunit alpha. Well, there is also a HEX-B gene that is closely related to the HEX-A gene. The HEX-B gene provides instructions for making the beta subunit of beta-hexoamidase A. So DNA from this HEX-A and this HEX-B gene are transcribed into this alpha subunit and beta subunit, and when they come together like this, they form something called an isozyme. And this specific isozyme is called isozyme A. And actually, a study done in 1995 in Ontario found that this isozyme A is the one that is responsible for the degradation of GM2 gangliosides. These GM2 gangliosides are the fatty substances that I mentioned earlier that need to be broken down. In this experiment, the scientists performed DEAE ion exchange chromatography to separate the molecules based on their surface charge. And what they found is that activity was 24-fold higher in alpha and beta cDNA transfected cells than in non-transfected cells. So our protein hexoamidase alpha has lots of moving parts, so let's break it down. We have our signal peptide, of course, that helps the protein go to the lysosome. There is also a propeptide that just ends off being cleaved during maturation, so that really serves no biological function. And then we have our long chain of amino acids that make up the part referred to the subunit alpha. There are about 3 sites for glycosylation, and this glycosylation is just kind of a quality control to make sure proteins aren't misfolded.
And so where should this protein end up in the cell, and how does it get there?
Hexosaminidase A is a lysosomal protein, so this protein follows a normal pathway of steps to get to the lysosome. This process takes place by translocation. The enzyme will be brought by a ribosome to the ER lumen. A messenger RNA must bind to a free ribosome. The signal sequence targets to nascent polypeptide and does compartmentalization in the ER lumen. The enzyme then emerges from the ribosome, and the signal sequence is recognized by the SRP. SRP binds to the ribosome, and then the signal sequence on the nascent polypeptide. This causes something called translation arrest and increases the SRP's affinity for GTP. This is now an SRP ribosome nascent polypeptide complex that now goes to the ER membrane. The protein gets there because of an interaction between the SRP and the SRP receptor present on the ER membrane. The protein then goes to the translocon. When the ribosome is attached to the translocon, the signal sequence is recognized. the growing peptide goes to the ER lumen. When a stop codon is reached in translation, the membrane-bound ribosome is released, and then this protein is transported by the mannose-6 phosphorylate to the lysosome. In a study done in 2003, purified hex A and hex B proteins from human placenta and crystallized it into a native lysosomal form. The crystal structure was determined by using MIR, which stands for multiple isomorphous replacement. Crystals were soaked overnight in solutions, and data was collected from the native and heavy atom crystals. And when they were presented, the crystal structure for hex A and hex B found that the phosphotransferase phosphorylates N-linked oligosaccharides of lysosomal proteins so that they can be subsequently recognized by the mannose-6 phosphate receptor and redirected from the Golgi to the lysosome.
So this protein is brought to the lysosome. Then does it do something?
It sure does. When hexoimidase subunit alpha enters the lysosome, it has a very important job. It removes the N-acetylglucosamyl residue from the fatty acids I mentioned earlier, the fatty acids that are GM2 gangliosides. And to maintain proper function, hexoimidase subunit alpha needs to hydrolyze these GM2 gangliosides to break them down. And what we see in Tay-Sachs is the buildup of these GM2 gangliosides to toxic levels. So the long process of getting this enzyme to the lysosome and the breakdown of these GM2 gangliosides still happens in someone with Tay-Sachs. However, it is not happening enough. It is happening so rarely that we see so much of this buildup. And so evidence we have for the incorrect protein function is looking at the buildup of these GM2 gangliosides and how it affects the body. Electron micrographs in one study showed that effective brain tissue revealed cytoplasmic membrane-like structures formed by the massive accumulation of GM2 gangliosides in the lysosome. And CAT scans and MRIs may be done also. A CAT scan of the brain in someone with Tay-Sachs shows the increased density of the basal ganglia. An MRI demonstrates the diffuse white matter changes. In another study with 3 children who were diagnosed with Tay-Sachs, their brains were looked at by spectroscopy, and this spectroscopy findings shows demyelization and neuronal loss.
So in someone with Tay-Sachs, would we see more accumulation of these GM2 gangliosides in the lysosome?
Exactly, Rachel.
I don't know much about the lysosome. Can you give me a brief overview of what its function is?
Of course I can. So the lysosome is a membrane-bound organelle found in all eukaryotic cells, and lysosomes are essentially killing and recycling centers. So their goal is to rid cells of waste products.
Okay, so you've talked about these GM2 gangliosides a lot, but what are they and what do they need to be broken down?
Thanks for bringing that up. This is a dense question because actually the functions are not completely understood. Studies done have shown that they have effects on concentration of calcium ions at the membrane surface and binding of toxins. It seems like they have pivotal biological functions for the central nervous system. So it seems like they need to be present at some point in development, but then they need to be degraded.
So, I feel like I know everything about Tay-Sachs now, but I'm dying to know, is there a cure?
I wish I had better news for you, Rachel, but right now there is no cure for Tay-Sachs. But of course, scientists are always researching and thinking of ways to correct things. So I found an interesting study that took cells from a terminated pregnancy, and these cells were shown to have a deficient hex A activity and accumulation of GM2 gangliosides. And further testing revealed that these were in fact cells that would have led to a diagnosis of Tay-Sachs. So the scientists' approach here was to transfect the Tay-Sachs neurological cells with solidase cDNA and to assay the cells for clearance of GM2 ganglioside. So what they did is they made cDNA that encodes for a lysosomal solidase. And solidases are normally found in the lysosome, and they catalyze the removal of terminal silylic acid groups. And so in order to later detect the silylidase, a vector was prepared with a polyhistidine-tagged human silylidase cDNA that could be detected with an antibody. 2 days after transfection, the cells were fixed and immunolabeled with anti-GM2 and anti-polyhistidine tags. What they found is that high levels of silylidase show virtually complete depletion of their accumulated GM2 gangliosides. So all of these cells that were positive for the transfected silylidase, which was 270 cells, showed complete or near reduction of GM2 gangliosides. So reading the study made me really happy because they were able to conclude that increasing the level of lysosomal silylidase in Tay-Sachs neurological cells is sufficient to facilitate degradation of accumulated gangliosides. So if we had something like an allosteric activator of silylidase, this could be one way to degrade GM2 gangliosides even if there is a mutation in the hex A gene that is preventing the beta-hexoimidase alpha subunit protein from doing this.
Wow, that is really inspiring news, Isabel. Thanks for answering all my questions today. I have a better grasp on Tay-Sachs now.
You're welcome, Rachel. Thanks for joining me today, and good luck with everything. Well, listeners, thanks for tuning in for another episode of Investigating Rare Diseases. I'm your host, Isabel, and I'll be back next week. Take care.